Hello everyone. So in this session we will discuss about the amino acid metabolism. So let me share the uh, content for you. Just hold on for a minute. So here we will start with amino acid oxidation and production of urea. This metabolism. So previously we shifted from biomolecules to the metabolism. We discussed different biomolecules, carbohydrate, protein, lipid, right, nucleotide, nucleic acid. Then we covered sugar metabolism, carbohydrate metabolism, which we took two sessions for that, which included glycogen metabolism, glycolysis, gluconeogenesis, which is anabolism, and glycolysis is a catabolism. Then pentose phosphate pathway, and then we also discuss about TCA cycle, then how the pyruvic acid which is produced from the glycolysis, how does it enter into the mitochondria, and then how it will enter into the TCA cycle in mitochondrial matrix, which will eventually produce the CO2 and ATP, which is energy source, and also it will produce NADH and FADH2. So as we discussed that in so last class we talked about that NADH and FADH2 which are the reduced form, so they can donate electron in the electron transfer chain, and then those electrons can flow through the electron transfer chain, right, which will initiate a proton motive force or PMF, right, which drives ATP synthesis in the inner mitochondrial membrane. So it will drive the ATP synthesis to produce ATP, right? So that we will discuss in today through our so actually that we will discuss in uh, oxidative phosphorylation chapter in this session only. Right. So so carbohydrate we did metabolism. So now we will and also we discussed fatty acid lipid metabolism. The fatty acid beta oxidation we did, right. So now we will talk about the amino acid metabolism, right? How these amino acids which are uh, ingested in our blood by ingested dietary protein, and then how these amino acids gets oxidized, and what is the fate of this amino acid oxidation, and how will they lead to production of urea, that we will see, right? So this is the overview of amino acid catabolism in mammals. So where you can see that intracellular protein which are getting by the dietary protein, right? So that will be broken down into amino acids, right? So both intracellular protein and dietary protein, both of them will be broken down to amino acid. These amino acids will uh, produce the, because as you know that amino acid, if you remember the amino acid uh, backbone, that it has a CH and H3, C double O minus, and R group, right? So if we remove the ammonium group from this, right? For example, if you know, for example, if we try to just draw amino acid backbone, So So if this is your amino acid, which is where is a cardinal carbon is there, which is attached with hydrogen, right? Which is attached with uh, NH3 plus, right? NH3 plus, and it has one hydrogen with this. Carbon has 4 valency. It has hydrogen. It has an R group, and it has C double O minus acid group, right? So now yeah. So now, if we remove this amino group from here as in form of NH4 plus, which is ammonium ion, right? As in form of ammonium ion, if you remove this amino group from here, then you will be left with this portion, right? This portion you will be left with CHR and C double minus, which is a carbon backbone, right? So after removing this ammonium ion, you will be left with this carbon backbone, right? So yeah. So now you see that these amino acids will be broken down into ammonium ion and carbon skeleton. And these ammonium ions will be used for the biosynthesis of the amino acids, nucleotide, biological amines, right? Or this this ammonium ion will combine with carboxyl. It will be converted into carboxyl phosphate, which will enter into the urea cycle. Finally, the cycle will produce urea as nitrogen excretion product. Right? Again, this intermediate of this urea cycle can be participated in the argi- aspartate argininosuccinate shunt of citric acid cycle, right? Through which it will enter into the citric acid cycle, for example, alpha ketoglutarate. Right? This intermediate is a common of citric acid. So when this will enter into the citric acid cycle or TCA cycle, which we discussed in the last class, it will produce CO2, H2O, and ATP, right? And from the citric acid cycle, the intermediates can be used to, for example, oxaloacetate is the intermediate of the CTC cycle, as we know, right? From our last session. So this oxaloacetate can be converted into glucose. We also discussed that how is it possible that this oxaloacetate will be converted into glucose by gluconeogenesis. So actually, it can be converted into malate in the cytosol, and the cytosol PEP carboxykinase will come and it will convert it into PEP. Then it will follow the gluconeogenesis. So that's how this oxaloacetate can be used as a source of glucose, right? Which is synthesized by gluconeogenesis. So this is the, and from this carbon skeleton, alpha keto acids will also enter into the citric acid cycle. For example, alpha ketoglutarate that will enter into the citric acid cycle and it will produce CO2, H2O, and ATP. Right? So this is the overview of the amino acid catabolism in mammals. There are different groups of amino acid uh, catabolism. For example, if the cellular protein, this is your cellular protein, if it is broken down to uh, amino acids, then this will be your amino acid. It will, to liver, this amino acid will react with alpha ketoglutarate, so it will transfer its amino group to the alpha ketoglutarate, right? So there will be a amination of this alpha ketoglutarate here, right? You can see. So this amino group will come from the amino acid which is coming from the dietary protein or cellular protein, right? So from that amino group is coming to alpha ketoglutarate to make it glutamate, right? This is amination. So this amino group is transferred. So that's why it is called transamination. And after transferring this amino acid will be converted into alpha keto acid, right? C double O minus, C double one O, and this R. And example is alpha keto glutarate, right? So now these amino acids, for example, alanine from muscle, that we will discuss briefly. This alanine from muscle will come to liver. In liver, this amino acid will react with this alpha keto acid, which is produced by this transamination reaction. So it will react with the alpha keto glutarate, right, to make pyruvate and glutamate. So basically, this will combine and it will make pyruvate and glutamate. Okay. So again, this glutamate can be converted into alpha keto glutarate in the liver by deamination reaction, where the amino group will be removed. That is called deamination reaction. So that's how this glutamate will be converted into alpha keto glutarate, right? And this ammonium ion. So this ammonium ion can be excreted from the cell by as a form of ammonium ion or urea or uric acid, right? If it is removed in the form of ammonium ion, right, then those animals will call ammonotelic animals. They are mostly aquatic vertebrates, for example, fish, bony fishes, right, and larvae of amphibia. They do the excretion in the form of ammonium ion, right? But for us, this ammonium ion enter into the urea acid, enter into the urea cycle to produce urea, right? So this ammonium ion can be converted into urea by this urea cycle. So for us, we do the excretion in the form of urea. So that's why we are called ureotelic animals, right? Mainly terrestrial vertebrates and also sharks. And if we, yeah, and if ammonium ion is releasing from the cell as in a form of uric acid, then they will be uricotelic animals, for example, birds and reptiles. Right? So if it is excreted in a form of uric acid, this is the structure of the uric acid, then those animals will be called as uricotelic animals. For example, birds, reptiles, they are uricotelic animals, right? So these are the fates of ammonium ion. In these three forms, it can be excreted, right? And as we saw that alanine from muscle can be converted into alpha this pyruvate and glutamate, right? And this glutamate will eventually lose the ammonium ion, and which will be excreted in the form of either iron or urea or uric acid. Similarly, if glutamine from muscle and other tissues, that will enter it into the liver, where this glutamine will uh, remove will remove their amino group in a form of ammonium ion, right? and then this glutamate will be converted into glutamate, right? When it will lose the amino group from the R group. So after losing this amino group, it will be converted into glutamate, right? And then again the glutamate can also be converted into alpha ketoglutarate by deamination reaction, where it will also lose its amino group from this from here, right, which came from the dietary protein. So with that will be excreted in the form of ammonium ion, right? So this is the amino group catabolism, different type of catabolism, right? If it is from alanine from the muscle cells, then this alanine will be react with alpha keto acid, for example alpha ketoglutarate, to which will give you the pyruvate and glutamate, right? Pyruvate means CH3 C11O and this C minus, right? And glutamate means amino acid, where R group is CH2 C minus, right? So that's how the amino group catabolism happens. So now let's see the part of human digestive gastrointestinal tract, right? So how the protein from the dietary fat and so the protein and amino acid from the dietary fat, how they get digested and how they get absorbed, right? So when the ingested food will enter into the stomach, then the stomach, as you know, the stomach has low pH, right? Because it has the uh, HCl secreting cells, and also the stomach has peptic cells which can produce pepsin, right? Or they will first produce pepsinogen, which is a precursor of pepsin, which can be eventually activated to form pepsin, right? Yeah. So, so the stomach, it can secrete pepsin enzyme, and it can has a HCl secreting cells, so that it will always maintain a very low pH, right? So this is a gastric gland in the stomach lining. If you zoom out the stomach lining, right, this is a gastric gland which has the parietal cells which will secrete hydrochloric acid, which I talked to you about, right? And it has chip cells here. These are chip cells. They can secrete pepsinogen, this one, right, which can be converted into the active pepsin. And there is gastric mucosa. Those gastric mucosal cells, they will secrete gastrin, right? So this is your stomach. This is the pancreas. In the pancreas, there is a exocrine cells. The pancreas. So, so here this is the rough endoplasmic reticulum. There are some zymogen granules and collecting duct. Okay, and then there is intestine, where the pH is converted to 7. So this pancreas will release enzymes. For example, as you can see that there are some zymogen granules are there, right? So they can secrete the zymogens from these granules, right? And they will be coming into the intestine, right? So in the intestine, the zymogen will be converted into the active protease, and they can chop out the proteins or the small peptides to amino acids, right? Or it can also chop a long polypeptide chain into small peptide chains, right? So in the intestine lining, there is villi. So the villi, uh, in the singular, it is called as villus, and it will right. So they will absorb the nutrient, amino acid or whatever digestive products are producing here. They can digest them. They can actually absorb them, and they can send them into the blood flow, right? So and it can also secrete the intestinal mucosa, which will absorb the amino acid through the help of villi, by which the amino acids, right, which are produced in the total digestion system, these amino acids will be mixed into the bloodstream. Okay. So now we will first in this entire protein amino acid digestion chapter, we will first talk about the reaction which is transamination reaction. The enzyme which is involved is aminotransferase that will transfer the amino acid from the dietary protein to alpha ketoglutarate. So this amino group will be transferred to alpha ketoglutarate to form glutamate, right? And this amino acid will be converted to alpha keto acid after losing this amino group that we talked about here. That in the liver, these amino acids which are coming from dietary protein, that will be converted into alpha keto acid after losing this amino group, and this amino group will be transferred into alpha ketoglutarate, right? So that it will become glutamate, right? So that's why it is called transamination reaction. That is only shown here, and the enzyme which is involved is aminotransferase. This is the first and this is the first reaction which happens in the amino acid oxidation phase when it comes into the liver, right? So from this amino acid you are getting glutamate and alpha keto acid. So and here it uses PLP as a cofactor. PLP which means pyridoxal phosphate, which is the prosthetic group for aminotransferase, right? So prosthetic group is the group which is conjugated with the enzyme, which is a non-protein part, right? So that is your prosthetic group. So similarly, PLP is also a prosthetic group. It is a non-protein part, right? But it is conjugated with the enzyme so that enzyme can do its work. Right? Then we saw that glutamate, for example, here if you want to again recapitulate this, that after this transamination reaction you are getting glutamate, right? So this glutamate can be converted into alpha ketoglutarate and ammonium ion, right? So yeah, so here also first glutamate will so in the first step, which is a reversible reaction, in that reaction glutamate will be oxidized by using this NADP plus uh, cofactor, which will accept the electron. So this glutamate will be oxidized, right? Here the single bond will become double bond here, and because it will lose one hydrogen here, right? So that's why oxidation. And then in the next step there will be addition of water, so which will remove this NH2 plus in the form of NH4 plus ion, and after losing this the H2O is 2 hydrogen and 1 oxygen, so 2 hydrogen will join with here, so it will become NH4 plus, and the oxygen will join here, right here. So it will become NH4 plus, and this will become, after accepting this oxygen, it will become C double minus, C double but O, then CH2 CH2 C double minus, which is alpha ketoglutarate, right? So this glutamate will lose this amino group, right, which actually came to the right. So this amino group is coming from the dietary protein, right? The amino acids which are evolved from the dietary protein. From that, this amino group came to glutamate. Now, when the glutamate will be oxidized by glutamate dehydrogenase in the liver, then it will produce the alpha keto acid, which is alpha ketoglutarate, and ammoniamine, right, which we saw here, right, here. That this dietary protein amino group from the amino acids is transferring into the alpha ketoglutarate to make glutamate, and then this glutamate is oxidized to form alpha ketoglutarate, and it will removing this ammoniamine. So that step is carried out by glutamate dehydrogenase, right? So that will ultimately produce the ammonium plus ion. Okay. So now we will discuss this step where we saw that alanine from the muscle tissue, how they are coming here in the liver and how they are contributing to the body. So, so if you see that alanine transports ammonia from central muscle to liver. So in the muscle protein, when it will be broken down to amino acids, then it will uh, give you ammonium ion, and then from ammonium ion to glutamate, right? Also in the muscle tissue, there is occurrence of glycolysis. So in the glycolysis, glucose is producing pyruvate, right? So this pyruvate is combining with the glutamate, right? Then it will becoming alanine and alpha ketoglutarate by alanine aminotransferase, right? So what is so what's happening here? The amino group is transferred from the glutamate or glutamic acid to pyruvate, right? So now the glutamate or the glutamic acid is converted into the its corresponding alpha keto acid, which is alpha ketoglutarate, and after amination, the pyruvate will be converted into alanine. Now this alanine will be released to the bloodstream. So now this blood alanine will come to the liver. The liver alanine will combine with alpha ketoglutarate to make again this just opposite reaction to make pyruvate and glutamate, right? And as we know that in glucose in liver there can be possibility of gluconeogenesis. So this pyruvate can be uh, participated in the gluconeogenesis, where as you know that pyruvate will be first converted into oxaloacetate and then PEP, and then the entire gluconeogenesis pathway will be started. So this pyruvate will be converted into glucose, right? And in the glucose, after being converted into glucose, this glucose will be released into the blood, right? So that it can rise the level of uh, glucose in the blood, right? So yeah, so that is the fate of the pyruvate in the liver, and in the glutamate, as you know that glutamate by in the reaction of uh, glutam oxidation by this glutamate dehydrogenase. So this glutamate will lose this amino group and it will be converted into ammonium ion and alpha ketoglutarate. Similarly, here also glutamate will be converted into the ammonium ion, which will enter into the urea cycle to produce urea. Right? So that's how the alanine can transport ammonia from the skeletal muscle to the liver. So that means in the liver, the one option is uh, transferring the amino group from the dietary protein, right, or dietary amino acid. Another option is from the muscle protein, the amino acids are produced. That amino acid can be also uh, in the muscle. That amino acid will be converted into the ammonium ion, right, by that particular amino acid dehydrogenase, right? Now this yeah. So now in the muscle tissue only, ammonium ion will be converted into the glutamate, right? When it will react with the alpha ketoglutarate, so it will be converted into glutamate. So the glutamate is combining with the pyruvate by alanine amino transferase. So from the glutamate, the amino acid is transferred to pyruvate. So pyruvate is becoming alanine, and the glutamate, after losing the amino group, it is becoming alpha ketoglutarate. Right now, this alanine can be transported to glucose to the liver, where again the just opposite reaction in the liver there is alpha ketoglutarate, right, which can be formed by how when the dietary amino acids, when they will lose the amino group, then they will be converted into the alpha ketoglutarate. So alanine will react, react with that, donates its amino acid to the alpha ketoglutarate, so it will become glutamate, and alanine will become pyruvate. And then from the pyruvate, it can produce glucose by gluconeogenesis in liver. This glucose will be released into the bloodstream, right? Then this, and through the bloodstream, that glucose can be received by the to the muscle tissue, where it can be used for glycolysis for producing energy, right? So this is a cycle. Again, from the glycolysis, it will convert the glucose to pyruvate. Then again the same thing. The second cycle will begin, right? So that is called glucose alanine cycle by, right. So here actually alanine serves as a carrier for ammonia, right? This alanine is a carrier of ammonia which is coming to liver, donating its amino group to alpha ketoglutarate, right? which is making glutamate. Then glutamate will be uh, oxidized to make alpha ketoglutarate and ammonium ion. Now this ammonium ion is excreted in the form of urea when it enters into the urea cycle. That's why uh, alanine is serving a carrier for ammonia, right? And because it is donating the amino group to uh, alpha ketoglutarate, which is eventually being converted into glutamate, right? Which is actually producing the ammonium ion. So that's why it is serving a carrier, right? And it is also serving a carrier of the carbon skeleton of pyruvate from skeletal muscle to liver, right? Because this alanine, after losing the amino group, right? After losing the amino group, alanine is being converted into pyruvate, right? Now that's why it is serving a carrier of the carbon skeleton of the pyruvate form of skeletal muscle to liver, right? Right, because this pyruvate is being used in the liver to form glucose by gluconeogenesis. That glucose, that same carbon skeleton is being embedded into glucose, right? And then this glucose is the same. I mean, the glucose which has the carbon skeleton from this pyruvate, which is coming from alanine, muscle alanine. So that glucose is again going back to muscle tissue for being used for glycolysis to produce energy in the muscle. Right? So that's how this glucose alanine cycle works, which in which alanine transports ammonia from muscle to liver, right? And this alanine is also giving, right? It is giving the ammonia to alpha ketoglutarate, right? And then it is also serving as a carbon backbone for the glucose which is being returned. By how? This, after losing this amino group, the remaining thing is carbon skeleton, which is pyruvate. Then it is being converted to glucose. The glucose is returned into the muscle tissue. So that's how it is serving carrier of both ammonium ion and the carbon skeleton, right, of pyruvate, right. So next, if we see that, okay, so now we saw that how the, so we actually discussed this figure, that how the, I mean in liver, how the amino acid oxidation takes place. So there are possibilities are uh, from dietary protein, the amino acids we are getting. So that amino acid is converted into, it is transferred into alpha ketoglutarate, which is a TCA cycle intermediate, right? So as a result, alpha ketoglutarate is becoming glutamate, and that amino acid is becoming alpha keto acid or alpha ketoglutarate. So that will react with the alanine which is coming from the muscle, right? So now this alanine is reacting with this alpha ketoglutarate to form, in the liver it is forming pyruvate and glutamate, right? Right. So this, so basically alanine is transferring this amino group to this alpha ketoglutarate in the liver, right? So this alpha ketoglutarate is converted into, after getting the amino group, it is converted into glutamate, and this alanine, when it is releasing this amino group, after releasing this alanine is being converted into pyruvate, right? Now this pyruvate, as we know that this pyruvate in the liver, it will be used for glucose production by gluconeogenesis, and then again this glucose will be sent to the muscle for glycolysis, right? This is one way. And yeah, and so this is the two ways for producing the glutamate here, right? One is for dietary protein amino acids, right, which is transamination reaction, right? That's how we can get glutamate. And another thing is from alanine, which is coming from the muscle. That is also transferring the amino group to alpha ketoglutarate, right, which will making glutamate here, and of course pyruvate, which is being used for the gluconeogenesis, as we saw that, to produce glucose, right? So these are the two ways to produce glutamate in the liver. Third way is glutamine from muscle and other tissues, so that this glutamine from muscle and other tissues will enter into the liver. So after this deamination reaction, this amino group in the glutamine side chain will be released as a form of ammonium ion, and it will be converted into glutamate after losing the amino group. It will losing the amino group and it will be converted into glutamate. Right? So these are the three ways of forming glutamate in the liver. Right? One is the alanine from muscle, one is from dietary protein amino acids, and glutamine from muscle and other tissues. They are forming glutamate. Then this glutamate is going through uh, deamination reaction by glutamate dehydrogenase, which is forming alpha ketoglutarate and the ammonium ion. Right? So that is the so that's all for the amino acid oxidation in the liver. Right. So now this ammonium ion, which is the end product in the amino acid oxidation of in the liver, so that will be excreted in the form of either ammonium ion for this ammonotelic animals, or for urea, or for uric acid. So when this ammonium ion will enter into the urea cycle, it will produce urea. Right? When it will enter into the uric acid cycle, it will produce uric acid cycle. So we saw that how from muscle and from other tissues and from dietary amino acids, right, how they are producing ammonium ion. And now we will see next that how these ammonium ions are excreted in the form of urea or uric acid. Okay. So yeah. So in order to be part of nitrogen excretion and to be excreted in the form of uh, urea, for that the released ammonium ion in the liver, which is produced from the amino acid oxidation, that has to enter into the urea cycle, right, which is occurring in the mitochondrial matrix. So now as we discussed that we saw how in the liver ammonium ion is produced. So now we will see how they will enter into the urea cycle to produce the final excretory product, which is urea, right? For and that is for the ureotelic animal. For ammonotelic animal, they don't have this urea cycle, so ammonium ion directly can be excreted, right? So the amino nitrogen can be excreted in the form of ammonia, but for the ureotelic animals, they will excrete the amino nitrogen in the form of urea, right? Like for example, us. And for the birds and the reptiles, they they do the excretion of the amino group of the nitrogen in the form of uric acid, right? So now we will see how in our body ammonium ion which is produced in the liver, how they can be converted into the urea so that they can be excreted as the urea, right? So what happens that uh, the enzyme catalyzing these reactions are distributed in the mitochondrial matrix and the cytosol. Right, and this urea production by urea cycle that exclusively occurs in liver only, right? And then liver releases this urea to the bloodstream, and then it will be transported into the kidney where it is, right. So through the kidney the urea is excreted into the urine, right. So now we will see how this urea production is occurring in the urea cycle of liver cells mitochondria. So you will see that this glutamate for the extra hepatic tissue, as we saw in the last figure, that glutamate from the extrahepatic tissues, muscle and other tissues, amino acid from the dietary protein, and this alanine from the muscle. So again, to just revisit, alanine from the muscle will donates its amino group to alpha ketoglutarate in the liver, right? So that the alanine will be converted into alpha keto acid after losing the amino group, right, which is pyruvic acid here, and this right, and this and this alpha keto glutarate, after receiving this amino group, it will be converted into glutamate amino acid, right. Same for the uh, other amino acids which are coming from the dietary protein. So they will in the transformation reaction, they will react with alpha keto glutarate. They will donate their amino group to alpha keto glutarate, right? And then alpha keto glutarate, after receiving this amino group, will be converted into glutamate, and this amino acid will be converted into the corresponding alpha keto acid for alanine, which is pyruvic acid, right? So now the end product is the glutamate in both of the cases, and when the glutamine comes from the extrahepatic tissues, the glutamine when it will come into the mitochondrial matrix, then in mitochondrial matrix, glutaminase that will convert glutamine into glutamate, right? How? It will just remove, as we saw in that chart, that glutamine has, suppose if we want to see that here, or if you see this diagram again, glutamine which is coming from the extra hepatic tissue. So this in the R chain of the in the R side chain of the R group of the glutamine, it has a amino group, right? NH2. So it will lose this amino group, and it will be this amino group will be removed as a form of NH4 plus. So after losing this amino group in the from the R group of glutamine, glutamine will be converted into glutamate. It will lose this amino group, right? Then again glutamate will be in the transformation reaction. Uh, Glutamate will lose this, you know. Yeah. So then after formation of the glutamate, as we know that which is coming from this extrahepatic glutamine. So after this diamidation reaction, glutamate will form, right? Now this glutamate will be oxidized to form alpha ketoglutarate and this ammonia bind. So it will lose this ammonia, right, as a in a form of ammonia bind. So this glutamate which is coming from the glutamine of extrahepatic tissue, that will also eventually form the ammonia bind, right? So here also the same thing it is happening, that glutamine from the extrahepatic tissues is entering into the mitochondrial matrix. Then glutaminase is removing this amino group from the glutamine, right? So it will making glutamate, which is eventually after glutamate dehydrogenase treatment, it will forming ammonia bind. So glutamate producing from the alanine muscle and dietary amino acid, it is this glutamate is entering into the mitochondrial matrix, where the glutamate dehydrogenase will convert it into the ammonia bind and alpha ketoglutarate, right? And for the glutamate, which is coming from the extrahepatic tissues, they so in the matrix glutaminase will convert glutamine to glutamate and ammonia bind. So eventually, all of them are forming ammonia bind as the end product, right? And this oxaloacetate, which is a TCA cycle intermediate, that is also reacting with the glutamate by the enzyme aspartate aminotransferase. So here also amino group of the glutamate is being transferred to the oxaloacetate, so that this oxaloacetate will be converted into aspartate after receiving this amino group, and after losing this amino group, glutamate will be converted into alpha ketoglutarate. Right. So here NH4 plus ammonia ion is being produced by these sources, right? So now this ammonia ion will so now the carbamyl phosphate synthesis one, it will so it will, once you get this ammonia ion to bicarbonate ion minus, right, this HCO3 minus, so they will become carbamyl phosphate when ammonium ion will be conjugated with the HCO3 minus. Then this carbamyl phosphate, so now this carbamyl phosphate will enter into the urea cycle, right? So so till this part, so till this part, when the ammonia ion is converted into the carbamyl phosphate, this part is occurring in the mitochondria, right, which is converting glutamate to carbamyl phosphate. Now this will enter into the urea cycle, which is occurring both in the cytosol and in the mitochondria. So in the mitochondria, this carbamyl phosphate will react with ornithine to combine to make the citrulline in the mitochondria, which will leave the mitochondria and it will enter into the cytosol. Then this ATP hydrolysis will be happen, and they will make a intermediate, which is citrulyl AMP intermediate. It will remove to phosphate, right, and this AMP group will be conjugated to the citrulline, so which will make citrulline AMP intermediate. Then aspartate will come, right? Now this aspartate, from where it is coming? That here, right? Oxaloacetate, which is a TCA cycle intermediate, right? So from oxaloacetate, so it is reacting with the glutamate, which is produced in the liver from the alanine from the muscle tissue, dietary amino acids, and glutamate from the extrahepatic tissues. So this glutamate is reacting with the DC cycle intermediate oxaloacetate, which is forming the alpha ketoglutarate, where this glutamate is losing its amino group and it is transferring this amino group to the oxaloacetate, so it is becoming aspartate. Right now, this aspartate is coming to the cytosol, right, and it is entering into the urea cycle, where this aspartate is is reacting with the citrulline AMP, right? So that AMP is removed, and then this aspartate, this pink portion is conjugate. Yeah, this pink portion and this green portion means the aspartate portion. It is conjugating or it is combining with the citrulline, right? So now this is argininosuccinate, right? This is the succinate part, right? This is the succinate part and this is the arginine part, right? So that's why it is called argininosuccinate, which is formed by combining aspartate with citrulline AMP. After releasing of the AMP, it is becoming argininosuccinate. Then, yeah. So after that, it will release the fumarate. So this portion will be released, right? And there will be oxidation. So the succinate will be converted into fumarate, which will be released, and the remaining portion is the arginine, right? So arginine will be the remaining portion. Now H2O will come, right? With this hydration reaction, this H2O will come and this H2O will take this portion. So it will become NH2C, then again NH2, and then this O of the H2O will join here. So it will become NH2, C, NH2, NH2 and C double bond O, which is urea. That will remove, and if we remove this portion, then the remaining portion will be NH3, right? Because 2 H will be added here. So NH3, CH2 3 times, then CH, NH3, C double minus, which is actually on it, right? Because now this because now from arginine you are actually converting the arginine to ornithine, because you are removing this portion, right? This 2 amino group is removing by taking one oxygen from H2O in a form of urea, right? So that's why this ammonium ion, which is coming from the oxidation, right? So this ammonium ion, which is coming from the amino acid oxidation, they are actually removing here in the form of urea. So if you can see that this ammonium ion, if you see the structure, this ammonium ion, which is blue color shade, right, get conjugated with the bicarbonate ion by using right here, right? So which is carboxyl phosphate. This carboxyl phosphate, this NH2 group is is conjugating with the ornithine to make citrulline, right? So this, if you follow this or track this NH2, so from citrulline, the NH2 is still here. It is citrulline AMP. Then aspartate is coming and it is conjugating here, right? So this AMP will be removed and this will form a argininosuccinate, where this CNH here is conjugated with the this aspartate group, right, which is making argininosuccinate, right? So this is the amino group which is coming from the ammonium ion. Then after removal of this fumarate, then the remaining portion is the arginine, of course. Here is the ammonium ion, right? And then in the hydration reaction, this portion is removing as a form of urea, right? So it has, so the urea has the amino group, this NH2, which is coming from this ammonium ion, right? So again, briefly, that muscle tissue alanine, dietary amino acids, and the right, so they are forming glutamate in the liver cytosol, glutamate is coming into the mitochondria to form alpha ketoglutarate and ammonium ion, which is entering into the urea cycle, right, by attaching with HCO3 minus to form carboxyl phosphate, and then it will enter into the urea cycle, right. So this NH2, which is coming from this ammonium ion, right, and from glutamine, which is coming from the extra hepatic tissue, it is entering into the mitochondria in the liver. Then glutaminase will transfer this ammonium ion to, right. So yeah, so from the R group of the glutamine, the ammonium, the next group will be removed in the form of ammonium ion, and so it will become, after losing the amino group, it will become glutamate, right? And then again the glutamate will be also converted into ammonium ion by glutamate dehydrogenase. So that's how NH4 plus will be formed in the liver mitochondria, which will enter into the urea cycle to produce urea. So that's how this nitrogen, this blue color nitrogen, which is coming from, ammonium ion, so that's how this nitrogen is releasing in a form of urea by urea cycle, right? And here this TCA cycle intermediate oxidation is playing a role, because it is by aspartate amino transferase, it is uh, taking the amino group from the glutamate, right? So it is becoming aspartate. Aspartate is coming here in the TCA, in the urea cycle, right? So it is reacting with citrulline AMP. AMP is getting removed, and then it is combining with the citrulline to make argininosuccinate, right? And now, in the same way, the urea cycle will be going on, and it will remove this ammonium ion in the form of urea, right? So that is the case of ureatelic animals, which can excrete the ammonium ion in the form of urea here by urea cycle, which takes place in the liver, right? So now, as you already saw, that there is a link between the urea cycle and citric acid uh, okay, yeah. So there is a link between the urea cycle and citric acid cycle. And the link is yeah. So urea cycle, citric acid cycle, and this aspartate argininosuccinate shunt of the citric acid cycle. So how it is happening? So if you start from the urea cycle in the right, so as you know that when the carboxyl when the carbo yeah. So when the carboxyl phosphate is combining with the like ornithine, right, to make citrulline, right. So that part of the urea cycle is occurring in the mitochondria, and then this citrulline, through the intermediate of citrulline AMP, it is combining with aspartate, right here. So now how this aspartate is coming, right? So this is the junction point between the urea cycle and this shunt, that the citrulline is combining with the arginine to make the argininosuccinate, right? So now, so as we see that this is the common point between these two cycles. So how it is getting the aspartate, right? So the process is, in the citric acid cycle, oxaloacetate, right, which is a citric acid cycle intermediate, so that is combining with glutamate, so which is a transamination reaction. So glutamate will transfer its amino group to this, oxaloacetate, right? So that this oxaloacetate will be converted into, after receiving this amino group, it will be converted into aspartate, right? And glutamate, after losing the amino group, it will be converted into alpha ketoglutarate. So now this aspartate, after taking from when the oxaloacetate, the TC cycle intermediate is converted into the aspartate. This aspartate is coming out from the mitochondrial matrix, right? And then it is combining with the citrulline to make argininosuccinate in the urea cycle, which is occurring in the cytosol. Right? So that's how this aspartate is coming. So that is a very good link between the TCA cycle and urea cycle. So that's why urea cycle is a way dependent on the citric acid cycle, because the citric acid cycle can only supply the aspartate here, right? Now there is a aspartate argininosuccinate shunt of the citric acid cycle. So how this is occurring, that this argininosuccinate, which is a intermediate of the urea cycle, so this argininosuccinate in urea cycle, it is if you remember that in urea cycle in the cytosol, when aspartate is combining with the with the citrulline, then it is making argininosuccinate, right? Here there is the next step, this pink portion which is coming from the aspartate side. So this pink portion is oxidized and it is being removed as a form of fumarate, right? And the remaining part is staying here as in a form of arginine, which is contributing in the urea cycle. But what is the fate of this fumarate? So this fumarate, which is coming from the aspartate, this fumarate will be converted into malate, right? It will be converted into malate in the cytosol, right? By malate. Okay, so now this malate, which is actually hydration reaction, water will be added, so it will be converted into malate, and then this malate, which is a TC cycle intermediate, and it is a transporter, so it can enter into the mitochondria from the cytosol. So it will be entered into the mitochondria from so it will enter to the mitochondria from the cytosol, right? It will come here in the mitochondrial matrix, then it will again participate in the TCA cycle, right? And as you know that the next step is the formation of the oxaloacetate. When malate malate dehydrogenase, when it will oxidize malate, it will form fumarate, right? It will form oxaloacetate, right? So that's how it can again enter into the TCA cycle, right? And this fumarate also, one fate is this fumarate will be hydrated to form malate in the cytosol, and then malate will come back. Otherwise this fumarate will come back to the mitochondrial matrix, and as you know that fumarate is also a TCA cycle intermediate, so it will enter into the TCA cycle also, the mitochondrial matrix, right? So that's how this there is a link between the TCA cycle and the urea cycle through this aspartate argininosuccinate shunt. Okay, so again what is happening, that in urea cycle, when the citrulline is coming out from the mitochondrial matrix to cytosol, then aspartate is coming to be combined with the citrulline to make argininosuccinate. So how is the aspartate coming? In the TCA cycle, the intermediate is oxaloacetate. So here the oxaloacetate is combining with glutamate. So here what is happening, amino group of the glutamate will be transferred to the oxaloacetate. So oxaloacetate will be converted into aspartate, right, in the mitochondrial matrix, and then aspartate will come out from the matrix to the cytosol, where aspartate will enter into the urea cycle by combining with citrulline to make argininosuccinate, right? Then it will be continued in the arginine. And as we know that when the argininosuccinate will be cleaved in the urea cycle to form the arginine and fumarate, this fumarate will be released, and this fumarate will be converted into malate in the cytosol, right? Then this malate will come back to the mitochondrial matrix and it will enter into the TCA cycle. Otherwise, fumarate itself will come back to the mitochondrial matrix and it is a cycle intermediate, so it will enter into the citric acid cycle, right? So that's how. So this aspartate arginine succinate shunt, there is a link between urea cycle and the citric acid cycle. Okay, so that's all for the amino acid oxidation and the production of urea. So next we will start in this session only that about oxidative phosphorylation. So that is occurring in the mitochondria, right? So if you see the structure of the mitochondria, then there is outer membrane. After that there is inner membrane, and between outer and inner membrane there is a intermembrane space. Inside the inner membrane there is matrix, right? And matrix contains all the citric acid cycle enzymes, and fatty acid oxidation is also taking place here. So it has the fatty acid beta oxidation enzymes, right? Amino acid oxidation enzymes also, we just studied that, right? And this inner membrane has this electron transfer chain complex 1 to 4, so that we will study where the electron should be flown from the NADH and FADH. Okay. Yeah. So now we will see that how the NADH, FADH2 which are produced from the glycolysis, fatty acid beta oxidation and TCA cycle. So how are they contributing to produce energy, right? now we will see how this NADH, FADH2 are contributing to produce ATP, right? So as we discussed, mitochondrial membrane. In the inner mitochondrial membrane, there are the if you see, there are the respiratory chain, electron transfer chain carriers, complex 1 to 4, and ATP synthesis is also there, right? So now we will see how electron travels from NADH and FADH2 throughout these complexes, which is electron transfer chain, and how they will actually be accepted by oxygen, right, and which is actually associated with ATP synthesis. That we will see. So first we will start with the inner membrane complex. So this is the path of electron from NADH succinate, right? So this is showing how from NADH the electron is transferring throughout these complexes, right? So here first NADH, so the matrix NADH is there because TC cycles are occurring in the matrix, so that will produce NADH. So this NADH will actually be oxidized, right, to form NAD plus, and this electrons which is released by NADH will be travel through this FMN complex, iron sulfur conjugate, right, and then from there to complex 3Q, right, and from the fatty acyl CoA, when these fatty acyl CoAs are using FAD, so this is reduced to FADH2. This FADH2 electrons are also coming to this complex Q. So now we will see how they are traveling, right? So the first in the complex one, the NADH is releasing the electron, which is right. So these electrons are received by the famine mononuclide FMN, right? And then it will travel to a series of iron sulfur center, right? And then it will be accepted by Q, where the Q is ubiquitinone, right? So this ubiquitinone will be reduced. They will accept the electron, and they will make 2 hydrogen, 2 proton from the cytosol. So it will become 2 proton, 2 electron. That means 2 hydrogen. So they will be converted to QH2, which is reduced ubiquitinone, right? So in this case, along with this incident from cytosol, from the matrix to the intermembrane space, right? Matrix is the inside, this intermembrane space is the P side. So from N to P, there will be a flow of 4 protons, right? Right, which is occurred by the proton flux, right, which is produces an electrochemical potential gradient, right, across the inner membrane, inner mitochondrial site, which is N side, that is negative matrix, and a P side, which is intermembrane space, which is positive, P for positive and for negative. So now that will create a potential difference between the matrix and the intermembrane space, right? So that's why the electric field will be there, which will pump out 4 protons from the matrix to the P side. Why? Because matrix is negative and P side is positive. So now the protons, right? So now so now it is against the electrochemical gradient, because this proton flux is actually, the direction is against the electrochemical gradient, because it is traveling from the negative to positive, right? From the negative to positive, a positive charge is traveling from negative to positive, which is against the electrochemical gradient, right? So but proton fluxes are still are able, they are actually forcefully, they are pushing these protons from the matrix to the intermembrane space, which is against the electrochemical gradient, which will actually conserve some of the energy, right? So these energies will be used to drive the ATP synthesis, that we will see. So in this step, the electron traveling throughout the complex 1, it is causing proton flux for proton flux, which is consisting of moving of 4 protons from the matrix side to the intermembrane space side or P side, right? Next, in the complex 2, what is happening? So this complex 1 is also known as ubiquitinone oxidoreductase, because here the ubiquinone is being reduced by accepting those electrons, right? Next, we'll see the complex 2, right? So this is your complex 2. Again, this is the matrix, right, which is N side. This is the periplasm, which is P side. So here the FADH2, which is product of TCA cycle and the fatty acid beta oxidation. So this FADH2 will release the electrons, so that the FADH2 will be converted into FAD, and these electrons will be traveled throughout this complex 2 by a couple through a couple of iron sulfur centers. Then ultimately it will be received by the ubiquinone in the inner membrane, right? Which is so right. So this diagram is for the complex 2 or the succinate deaminase for E. coli, which is a bacteria. So that's why there is no mitochondria, but this is similar because they also have the electron transfer chain complexes in the inner membrane of the cell, right? And they have the outer membrane also, so that's why they have the periplasm side, which is P side, and cytoplasm is the equivalent to matrix, which is N side. So here electron is traveling from FADH2 to ubiquinone, which is in the inner membrane. So here ubiquinone is receiving the electrons to make this QH2, which is reduced ubiquinone, right? So that is the complex 2, where also ubiquinone is reduced to QH2. So if you see from the complex 2, okay, so in complex 2, which is referred as the succinate dehydrogenase, so it will uh, so it will oxidize the succinate, so that the electrons released from the succinate will be finally accepted by ubiquinone to make QH2. Okay, now the complex 2. So now we saw that from complex 1 and complex 2, ubiquinone ubiquinone is reduced by receiving the electrons. Right, so that will produce QH2. Now how this from ubiquinone, which is QH2, how these electrons will be transferred to the cytochrome C, that is through complex 3. So here you can see that so again this intermembrane space is P side, and the matrix is N side. So this is the structure of the complex 3. So now if you see the how this is happening, that So this QH2, which is final product of complex 1 and complex 2 electron transfer. So this QH2 will release the electrons. So the electrons will be traveled from this iron sulfur center to cytochrome C1 and then to cytochrome C in one direction, and in other direction the electrons will travel from reduced ubiquinone to Bl and BH, right? So here so here the QH2 is reacting with the cytochrome C1, right? And then so what is happening? This QH2 is releasing electrons, so it will be oxidized. So that's why it is Q minus and one electron here. And then the cytochrome C1, which is accepting the electron, so it will be reduced, right? Cytochrome C1 will be reduced, right? And then in the second step, now the QH2 will be transformed into Q minus and this free radical, right? So again, when this Q minus, so again this Q minus and free radical, when they will release the electron, then this again it will the electron will flow throughout this path, and it will reduce the cytochrome C1, right? So which will actually so which will release of 2 HP plus, right, here in this direction from matrix side to the P side. It is again due to the proton flask, right? And in the second step also 2 protons will be pumped to the intermembrane side or P side. So the net equation is 2 molecules of cytochrome C1 will be reduced here if you add these two equations, right? And 4 protons will be traveling from the inside to the P side, right? Because of the proton flask, right? And this QH2 will be converted into Q. So in the first oxidation step, QH2 will release the electrons, right? So that it will right. So so QH2 is releasing the electrons. So this H2 means two protons and 2 electrons. So 2 protons will be coming out from the inside to P side, right? Right here, and this will be remaining as the Q minus with a free radical, right? That means it still has 2 electrons. One is in a negative charge and one is a free radical form, right? And then this form will actually right, and then when the second molecule of QH2 come, then it will, and already and already there was Q minus with this free radical. So when the second molecule of QH2 will come here, right, then again the same thing, it will release its electron throughout this complex 3. So it will release these 2 protons, right, and this will be converted to the oxidized ubiquinone, right, and then this Q minus also will take the protons from this negative side to Hn plus, and it will convert it into the QH2, right, because it already has 2 electrons. Now it will take 2 protons to form QH2. right? 2 proton and 2 electron means 2 hydrogen. So that's why it will be 2H2. So that is from negative side of the matrix, right? And because of this oxidation of the second molecule of QH2, so it will become actually the Q, and these 2 protons will be released into the p side. That's why 2 protons are released into the p side, and QH2 will be converted into Q, right? But in this case, in the first oxidation step, this Q, which is when the first molecule of KH2 will be oxidized, so it will release electron here, release 2 proton here, only the remaining thing will be Q, right? And then this Q will actually take this electron which is coming from this path, right? One single electron, and it will become Q minus with a free radical. What is happening here again? This Q is this QH2 is releasing 2 electrons, right? Because H2 means 2 proton, 2 electrons. So 2 protons is coming out to the p side, that's why 2Hp plus 2, right? And 2 electrons. Among them, 1 electron is going in this direction to reduce cytochrome C1, right? And the second electron is going here in this direction, right? So that throughout these carriers, it will actually reduce the Q, right? Which is this Q, ubiquinone, which was produced by QH2 electron release. So this electron will again reduce this ubiquinone here, which was produced from QH2. Then it will become Q minus and with a free radical, right? Which has now in the next step of, right? So that's why this QH2 is ultimately producing Q minus with a free radical, right? And this cytochrome C1 is reduced. In the second step, when the second molecule of QH2 is releasing electron, so then again same thing. H2 means 2 proton and 2 electron. So 2 protons are coming out to the p side, right? 2 electrons are coming throughout, right? Now remaining is the 2 electron. 1 electron is going through this path, so it will reduce the cytochrome C, right? Cytochrome C1, so it is reduced, and then another electron will travel throughout this path, and it will reduce ubiquinone, right? So now it will reduce this Q minus which was produced in the first step of oxidation. So this electron will reduce this ubiquinone, right? And then so that, and it will take, so yeah, so now it is getting one more electron, right? So that means it is getting 2 electrons, and then it is taking 2 protons from the matrix, so that it will form QH2, right? 2 hydrogen molecules mean 2 protons and 2 electron, right? So again in the second step, QH2 is coming, it is donating electron, 1 electron is going through this direction, it is reducing the cytochrome C1, right? And upon this oxidation reaction, 2H plus is coming out to the p side, right? And remaining is the 2 electron. One is traveling through this path to reduce cytochrome C1. Another is traveling through this path, right? Another is traveling to this path to reduce the previously produced Q minus with a free radical, right? Now there is only one free radical, right? So now it will, when it will accept one more electron, right, then it will become Q minus minus, it will have 2 electron, and it will take 2 proton from the n side, matrix side. So it will become, right? So that means 2 proton, 2 electrons means 2 hydrogen atoms. So it will become QH2. So finally it will become QH2 here, right? And this Q minus will be reduced to QH2, and this QH2 will be forming this Q, right? And the cytochrome C1 will be reduced, right? and it will release two proton to p side, and it will take two proton from the n side. So the overall reaction will be, if you add these two equations, then it will be QH2, 2 cytochrome C oxidized plus 2HN plus, which will give you ubiquinone Q, and 2 cytochrome C1 reduced, and then going out of 4H plus in the P side. Right? So it is taking 2 hydrogen from the N side and it is releasing 4 proton in the P side. That is called Q cycle in the of the complex 3. Right? So that's how in this cycle, in this complex also, the cell will pump out the proton against the electrochemical gradient from negative to positive. Right? Again, it will conserve some energy because of this, opposite to the, as they are pumping proton opposite to the electrochemical gradient, so it will be making energy, it will conserving energy because of this process, which will be used for the ATP synthesis. Right? In the complex 4, you will see that in the complex 4, so what we saw in this case, that that finally this QH2 is finally oxidized to ubiquinone, so it will can take part to the next round of the electron transfer chain, right? And it is reduce this cytochrome C1. Now the cytochrome C1 is reduced. Right? So now see, this cytochrome C1 is reduced. So now this cytochrome C, so now from through the cytochrome C1, the electrons are going to the cytochrome C, right? So now this cytochrome C, which are reduced, so they are 4 molecules of cytochrome C are releasing 4 electrons, right, which is traveling from, and now, so in this case you have to know that in the complex 1, as we saw, that electron is from NADH, electron is transferred to ubiquinone to make QH2, and this QH2 is mobile, so that's why it can travel through all this membrane and it can reach to the complex 3, right, which is cytochrome BC1 complex, where it can participate in the Q cycle, right? So, and in the Q cycle, ultimately this QH2 will reduce the cytochrome C. So now this reduced cytochrome C can travel, and it will travel throughout this membrane to complex 4, right? So so this is the complex 4. So when it will reach to complex 4, the cytochrome C will release electron. 4 cytochrome C will release 4 electron from the P side, positive side, internal space, and this electrons will be traveled from this CUA, right, and then this iron copper center, right, and then CUB, right, and then these electrons will be accepted by oxygen, which is the terminal electron acceptor. So oxygen will accept these electrons, right, as a terminal electron acceptor. So oxygen will be reduced by accepting the electrons. It will be reduced to form 2 molecules of H2O, right? So now oxygen means 2 oxygen atoms, right? Now it is taking 4 electrons. Now it will taking 4 proton from the inside. So 4 proton and 4 electron from this reduced cytochrome C, it is taking 4 electrons. So 4 electron from the reduced cytochrome C, and then 4 protons from the matrix, right? That means 4 hydrogen atom. So now this oxygen is attaching with the 4 hydrogen atom. So that's why 2H2. That means 4 hydrogen atom and 2 oxygen atom. So that's why it is giving 2 molecules of H2. Right? So that's why the oxygen is serving as the terminal electron acceptor, which is taking electron to be reduced to form H2O. Right? And in this process, again against the electrochemical gradient, 4 protons are pumped from the matrix to the inside matrix to the p side in intermembrane space. Right here, 4 protons are pumped. So again, pumping against the electrochemical gradient, so that will also consume some energy which will be used for the ATP synthesis, right? So that's how we saw that throughout the, electron transfer throughout this complex 1, 2, 3 and 4, there are some protons are getting pumped against the electrochemical gradient from matrix to electrochemical gradient at this direction. Sorry, from matrix to intermembrane space, from N to P side, right? So protons are getting pumped when electrons are transferred throughout this electron transfer chain. Right? So this is a summary. You can see that in the complex 1, NADH is oxidized to NAD plus. It is releasing electron, which is accepted by the ubiquinone, right? And it will pumping 4 protons. And in the complex 2, which is a succinate dehydrogenase, so it will oxidize the succinate, so which will make fumarate, and it will release electron, which will be again accepted by the ubiquinone, right? So the complex 1 and complex 2 will travel the electron, right? So that electrons can be accepted by the ubiquinone to make QH2, and among them complex 1 is associated with the proton pump, 4 proton pumping from the inside to P side. Now this QH2 will come to complex 3, where it will transfer its electron throughout this complex 3, which right, and finally those electrons will be accepted by the cytochrome C, right? In this process, the electron transfer throughout the complex 3, there will be 4 protons will be pumped from the inside to P side, right? Then the cytochrome C will reach to the complex 4, where cytochrome C will donate it electrons. It will release it electrons throughout the complex 4. So it will be transferred to the oxygen as the terminal electron accepted, right? And it will take protons from the N side to make H2O, right? And this process is associated with the for each molecule of the cytochrome C, there will be so actually for each molecule of production of the H2O, there will be 2 protons will be pumped from the inside to P side, right? So that is a summary of electron transfer chain, which the outcome is the production of the H2O, right? Where the oxygen is the terminal electron acceptor, and here a complex 1, 3 and 4, which are associated with the proton pumping from the inside to P side, which will conserve some energy. So now the energy which is conserved by this proton pumping, that will be used to synthesize ATP, right? This is called the proton motive force, because as you know that the protons are travel from the N side to P side against the gradient, right? Here, so as it is against a gradient, right? So that's why there will be a proton motive force which will bring back this protons from P side to N side, right? So, and this is important for the ATP synthesis. So for ATP synthesis, as you know again, that complex 1, 3 and 4, they are the consequence of the electron transfer is the proton pumping from the N to P side. So, as a result of this, as it is against the electrochemical gradient, then the matrix will be negative charge, and the intermembrane space will be positive charge. So that's why there is an electrochemical gradient from P to N, right? Plus to minus. So now these protons which were pumped, they can be brought back to the matrix by using this proton motive force, which is because of this electrochemical gradient, right? Now this energy is used for the ATP synthesis, right? And that is done by the ATP synthase. Okay, so if you see the ATP synthase, this is the ATP synthase complex, right? So yeah, this is the ATP synthase. So when the protons will be brought back, then the ATP synthase will use ADP and PI, right, to form ATP. Yeah, so that's how the electrons which are transferred throughout this electron transfer chain, they will create motive force which will actually uh, carry out the ATP synthesis by ATP synthase, right? And then this is coupled electron transfer and ATP synthesis in the mitochondria, that is coupled, right? So that can be very easily tested, that if you use experiment where you are measuring the O2 consumed and in this axis and ATP synthesis in this axis, then in the time gradient when you will add ADP and PI, right, you are adding succinate for the complex 2, right, then you will see after adding of the succinate here from this point, uh, yeah, so from this point you will see that oxygen consumed will be rapidly increased, because now the electrons are travel throughout this complex, and oxygen is reduced to water, right, which is actually conjugated, coupled with the ATP synthesis. Black one is the O2 consumed. So if you see O2 upon addition of succinate, complex activation, the O2 is consumed, okay, and along with that ATP is being synthesized, okay. But when you add an inhibitor for this complex, for this electron transfer chain, which is cyanide ion, so which is inhibitor of the electron transfer chain, so when you are inhibiting the electron transfer chain, then obviously O2 consumption will be reduced, will be stopped, and ATP synthesis is also stopped, right? So that's why they are coupled, right? Electron transfer chain, which is measured by O2 consumed, and ATP synthesis, they are coupled, right, in the mitochondria, right? But now, if you add some, if you use some uncoupling agent, then let's see what is happening. So here the same thing, the O2 consumed and ATP synthesis. So when you add a succinate and ADP and PI, then you know O2, this black line indicates the O2 consumption. So now the complex are activated. So they will electrons will be transferred and they will reduce oxygen, right? So that's why O2 consumption will be increased, and along with that ATP synthesis is also increased. But when you use an uncoupling agent, which is venturecittin or oligomycin, which are specifically ATP synthase inhibitor, they are not, right, they are not electron transfer chain inhibitor. So when you are inhibiting the ATP synthesis or ATP synthase, then you can see ATP synthesis is stopped. But along with that, the electron transfer chain is also stopped, right? Because oxygen consumption is reduced, the rate is reduced, right? So still now it is coupled. But when you use an uncoupling agent, for example, dinitrophenol, which is a uncoupling agent, so if you will add this, then these two things will be uncoupled. So ATP synthesis will be blocked because you have added the inhibitor. But DNP, which is uncoupled, is added. So that's why, although ATP production is blocked, but still electron transfer chain will be active because there will be O2 consumption, right? So if you add an uncoupler, then or uncoupling agent, then the electron transfer chain will be uncoupled with the ATP synthesis, right? Otherwise, normally they are coupled in the mitochondria, right? So yeah, so I think that's all for today. Okay, so we will so we discuss about the oxidative phosphorylation and the amino acid oxidation and how how does it get excreted in the form of urea. For that we discuss the urea cycle also, right? Okay, so that's all for today's session. We will again meet on the tomorrow for the next session. Okay, thank you for joining.